Welcome to This Explainer. Today, we are putting medical research on trial. We're stepping into the role of a clinical judge to really audit the math behind clinical studies. You know, when you're digging through medical literature, it is incredibly easy to get completely lost in a sea of numbers, percentages, and bold claims. But today, we're going to cut through all that noise. We'll break down the foundational architecture of a study so you can instantly know exactly what the statistics are actually telling us. Let's get into it.
Okay, so the very first question we have to ask ourselves, and this instantly tells us what kind of study architecture we're auditing, is this. Did you follow them forward or look back? Every single time you're presented with a clinical study, whether it's linking smoking to lung cancer or some trendy new diet to heart disease, this is the absolute first question you must ask. The direction of time literally dictates all the math that follows.
Here is our docket for today's proceedings. We'll be hitting the directional divide, the attributable risk signal, going through a live execution drill, and we'll wrap up with the executional parole.
Starting off with Section 1, the directional divide, relative risk versus the odds ratio.
So we have this 50-50 dilemma between relative risk and the odds ratio, and it all comes down to the flow of time. Cohort studies are forward-looking. They follow exposures into the future to see what happens. Case control studies, on the other hand, are backward-looking. They take a disease and look back into the past for suspects. Imagine a cohort study as standing at the starting line of a marathon and watching to see who actually finishes. A case control study is more like standing at the finish line, gathering up the runners and asking them what they ate for breakfast yesterday.
When we're looking forward, we use relative risk to figure out how much more likely an exposed person is to get a disease. Because we're following people from a state of health straight into disease, we can calculate true incidence. It's really just dividing the incidence in the exposed group by the incidence in the unexposed group.
Conversely, when we look backward, we use the odds ratio. This calculates the odds that a person who already has the condition was exposed to a specific risk factor. Because we artificially selected the number of diseased patients and healthy controls for our study, we actually can't calculate true incidence. Instead, we use cross-multiplication, A times D divided by B times C, to find the odds of exposure. Now, what's really fascinating here is a mathematical shortcut known as the rare disease assumption. If you're auditing a case control study on an incredibly rare condition, the odds ratio acts as this temporary bridge, closely approximating the true relative risk. It's a pretty fantastic shortcut built right into epidemiology. To audit for actual signal versus just pure noise, we absolutely must memorize the null value of 1.0. If a 95% confidence interval for a ratio crosses this number, there is no statistically significant association. It is purely noise. A relative risk of exactly 1.0 means the risk in the exposed group is identical to the unexposed group. The exposure literally did nothing. Moving right along to Section 2, the attributable risk signal, AR versus ARP. Moving beyond just associations, we hit the diagnostic dividing line to ask, how much disease is actually caused by this exposure? This brings us to the classic 2x2 grid. It's the foundational blueprint for calculating absolute differences in clinical trials. A represents the exposed who got sick, while C captures the unexposed who still got sick anyway. It's the perfect setup for our next calculation. Attributable risk strips away all that background noise to tell us the precise excess risk. We calculate it simply as the incidence in the exposed minus the incidence in the unexposed. We are literally just subtracting the baseline rate of disease from the elevated rate to find out exactly how much blame we can pin on the exposure itself. And if we take it a step further, calculating the attributable risk percent reveals the exact proportion of that disease we could completely eliminate if we just removed the exposure.
This is absolutely crucial to grasp. While a huge relative risk might sound absolutely terrifying in a headline, a common exposure with a high attributable risk is what actually impacts public health. For example, a rare chemical spill might have a massive relative risk of 20, but maybe it only affects 5 people. Meanwhile, a super common dietary habit might have a tiny relative risk of 1.2, but because millions of people are exposed, the attributable risk translates to thousands of preventable hospital visits.
Let's move to Section 3, the live execution drill. Time to put these formulas on trial. Okay, let's dive into a simple 3-step algorithm that turns complex biostatistics into a totally straightforward, logical process. By forcing yourself to systematically identify the architecture first, you completely eliminate the chance of using the wrong formula. Step 1, identify the architecture. Step 2, pick the formula. Step 3, execute. Let's look at some specific study data on smoking and lung cancer. Imagine we have a scenario where 10% of smokers develop lung cancer, compared to just 2% of non-smokers. We know the exposure is smoking, and we are looking at the outcome, which is lung cancer.
Because we're following the exposure forward in time here, we calculate relative risk. 10% divided by 2% is 5. So what does that actually mean? It means smokers are 5 times more likely to develop the illness. It's a clean, undeniable signal.
But what if we looked backward, starting with the cancer patients instead? In that case, we've used the cross-multiplication of the odds ratio. Let's say we take 60 exposed cases times 80 unexposed controls and divide that by 20 unexposed cases times 40 exposed controls. That gives us an odds ratio of 6. Same exact exposure, but a different study design means we need a different mathematical tool.
Now, think about calculating the attributable risk using some dietary data, like fish consumption. By simply subtracting the baseline incidence from the exposed incidence, we isolate the exact burden of disease. For instance, we might find a 0.05 excess risk caused specifically by that lack of fish in the diet.
Finally, Section 4, our executional pearl, the relative risk reduction versus absolute risk reduction trap.
We really have to examine this ultimate execution gap, because this is where pharmaceutical marketing often clashes hard with absolute statistical reality. Imagine a highly touted new drug that drops the risk of a stroke from 2% in the control group down to 1% in the treatment group. The intervention clearly works, right? But the way we talk about that tiny gap changes absolutely everything.
Here is the trap. A pharmaceutical company is absolutely going to market this as a staggering 50% relative risk reduction, even though the absolute risk reduction, the actual physical drop in risk, is a mere 1%. Going from 2 to 1 is technically halving the risk, so sure, it's 50%. But in the real world of patients, treatments, and side effects, you're only dropping the absolute event rate by a single percentage point.
As the source material explicitly warns us, relying solely on relative numbers can massively overstate the actual tangible benefit of a therapy. Think about it. If an ad tells you a pill reduces your risk of a rare disease by 80%, but your baseline risk was only 1 in a million, well, that 80% reduction doesn't actually mean much for your daily life, does it? Which brings us to the golden rule of the clinical audit. Never, ever make a clinical decision without calculating the absolute truth first. When you know the absolute risk reduction, you can confidently weigh the true real-world benefits against the costs and potential harms.
Next time you see a massive medical breakthrough touted on the news, ask yourself, are they selling you the relative risk or the absolute truth? Protecting yourself from statistical noise isn't just about passing board exams. It's about being an uncompromising judge of the facts in front of you. You now have the tools to audit the architecture, follow the direction of time, and calculate the real impact. Thank you for joining me on this explainer. Keep asking the tough questions.